Hello, I am Dr. Jafar Shirazi, and today's topic is antibodies. We will cover in this presentation the following topics: definition, then we will discuss nature, then we will go for structure, types, isotypes, features of individual immunoglobulin, their comparison, and in the last of this presentation, we will see clinical importance of the antibodies. Antibodies are basically known as immunoglobulins, represented by Ig. Their nature is glycoproteins, and these are produced by effector B cells, commonly known as plasma cells. Their main function is to recognize, bind, neutralize, and help eliminate pathogens. Each antibody has two type of chains. One of the type is heavy chain and the other type is light chain. The type of the antibody is defined by the heavy chain such as IgG, IgM, IgA, IgE and IgD, and the light chain may be of the kappa or lambda. Antibodies have two type of regions. One of the region is known as the variable region and the other type of the region is constant region. The other feature of the antibody is the presence of hinge region, which provides flexibility for antigen binding. It is present in IgG, IgA and IgD, but it is absent in the IgM and IgE. Two type of fragments regions are also present in the antibodies. First one is the FAB, fragment antigen binding, and the second one is FC, fragment crystallizable, interacts with immune cells and plays a very important role in the complement system. Antibodies have been classified into five types known as IgG, M, A, E and D, and they are very different in function and structure. First antibody we will discuss is the IgG. This is the most important antibody. As far as its structure is concerned, this is monomeric and has Y shape. This is the smallest in size, easily diffuse into the tissues, and their four subclasses have been identified as IgG1, 2, 3 and 4. When we see the features of the IgG, 70 to 75% of the total serum immunoglobulins is the IgG. This is the only antibody that can cross placenta, and the half-life is the maximum, that is 21 to 23 days. It can play its role in the activation of the complement system. It is the, you can say, the main antibody which plays its role in the secondary immune response. It can neutralize toxins, viruses. It is very important in the opsonization and provide passive immunity to the fetus, most important in long-term immunity and vaccination. Next antibody is the IgM. This is the first antibody which is expressed on the B cells. It is pentameric in structure. These five chains are joined by the J chain. It is very large molecular in shape. This is the, as far as the features are concerned, this is the first antibody produced by the naive B cells. It is excellent complement activator, and its role in the agglutination is very important. Its major function is to provide the primary immune response. It helps to detect recent infections, efficient in eliminating pathogen very early as the pathogens get enter into the body. This is the first antibody which can encounter with these invading pathogens. Next antibody is IgA. It is the antibody which is very important in mucosal immunity. As far as the structure is concerned, it is monomeric in structure, but uh, it can be monomeric when it is present in the serum, but it is dimeric when it is present in the secretions. The main feature of this antibody is that it is present in saliva, tears, breast milk, mucus, and respiratory tract, GIT, and urinogenital tract fluids. It is very resistant to proteolytic enzymes. The main function is to protect mucosa from infection, prevents pathogen attachment, provides immunity to infants via breast milk, important in GI, respiratory, and oral immunity. Next antibody is IgE, which plays very important role in allergy and parasitic infection. It is monomeric in structure. It is present in very low concentration in the serum, but has very high affinity to the Fc receptors on the mast cells. It is the mediator in the type 1 hypersensitivity reactions and causes different type of hypersensitivity reactions like asthma, anaphylaxis, and others. The last antibody is IgD, which can be observed as B cell receptor antibody. It is also monomeric in structure. It is present mainly on the surface of the naive B cells, low in concentration in serum. It acts as a B cell receptor and helps B cells activate and differentiate into plasma cells and has a very important role in the regulating immune response. We have a summary in the form of table among the 5 classes of the antibodies. The first one is the IgG, then M, A, E, and D. In the second column, the structure of these antibodies is given. IgG is monomeric, IgM is pentameric, Ig is dimeric, but when it is present in the secretions, IgE is also monomeric and same for IgD. Serum concentration is very high for the IgG and the lowest for IgD, and the key functions for IgD are the secondary immune response, opsonization, neutralization, for IgM, primary immune response, and the complement activation. IgA plays important function in the mucosal immunity, and the IgE plays important role in allergic reactions and in parasitic defense mechanism. And the IgD is basically acting as a B cell receptor. Uh, in the last column, the main features of these antibodies is given. For IgG, it can cross placenta and its half-life is the longest among the other antibody classes. IgM, this is the first expressed antibody on the B cells, and the next IgA is main secretory antibody in the breast milk. IgE, it can bind with the mast cells and basophils, and the IgD It is present on the naive B cell. Clinical importance of the antibodies is given one by one. The first clinical importance is in the diagnosis, then in the vaccination, and then in the autoimmune diseases. Last slide of the presentation includes the clinical importance. The fourth clinical importance of the antibodies is in the therapeutic form, which is mainly attributed to the monoclonal antibodies, which are used in the cancer therapy, autoimmune diseases, and then infections. In the brackets, the typical example of the antibodies is given. And also, antibodies have very important role in the allergies, uh, like IgE mediated hypersensitivity, anaphylaxis, diagnosis, and management. And the newborn immunity, IgG crosses placenta and protects newborn for 3 to 6 months. And thank you so much for watching and listening the presentation.